welcome to welcome to 13 Universal. Okay. Um, this is Shedro, learning beyond barriers, and we're looking at biological molecules. All right, biological molecules, um, part two. You can see. So, and in this video, we're going to be focusing on carbohydrates and lipids as soon as possible. So let's go, guys. You remember what I told you that biological molecules, basically, biological molecules, they are building blocks. You remember? Building blocks. Don't forget that. Building blocks of life. So they make up living things. Anywhere there's life, you'll find biological molecules there. Okay? So, for example, this is a biological molecule. This is an amino acid. Okay? This is glucose. Okay? All right. So, this is lipid, DNA. All of these are they are biological molecules. So, biological molecules, there are important facts I want you to know about biological molecules. So, facts, don't forget that. There are facts about biological molecules. Number one, biological molecules is that this is important that all biological molecules are made up of atoms of elements. Atoms of elements. Now, this is all of them. For example, if I have this is lipid. I have lipid here. I have carbohydrate. You know, I have protein. Okay, I have nucleic acid. Let's go, nucleic acid, and of course I have water. Quickly, so if you check what makes them up, you check the atoms of elements that make them up. Water consists of oxygen and hydrogen. Lipid consists of carbon, hydrogen, oxygen. Carbohydrate consists of carbon, hydrogen, oxygen. Protein consists of carbon, hydrogen, oxygen, nitrogen, basically. So nucleic acid consists of carbon, hydrogen, oxygen, nucleic acid. Okay, and phosphorus. So if you want to, form a fever, this is O, cho, cho, chon, chomp. Just remember that. Okay, that's an important fact. An important fact about all biological molecules. An important fact. Okay, so they are all made up of atoms. And another important fact about biological molecules, guys, as we go on, is that biological molecules, like we mentioned, the biological molecules, just like their name sounds, very importantly, they, apart from water, okay, they are macromolecules. So water is no macro, so they are macromolecules. All right, all biological molecules are macromolecules. Macromolecules, right? Macromolecules means they are large molecules, apart from water. So that means that you see, water is micro, it's small. So lipid, carbohydrate, protein, nucleic acid, they are all large molecules. And another thing is that among all of them, right, there are macromolecules. Of course, let's talk about macromolecules. I'm giving you general fact about biological molecules. All right. So macromolecules are usually made up of components, smaller, all right, subunits. They're always made up of smaller subunits, right? Very important. For example, you know, lipids are made up of glycerol, glycerol and fatty acid. Fatty acid. See, we check out carbohydrate made up of monosaccharides. Monosaccharides. Okay, and then protein made up of amino acid, and then nucleic acid made up of um, nucleotides. Nucleotides. Okay, very important. So now they are made up of small subunits. But if you check all of them, some of these macromolecules are made up of what we call similar. Some of them are made up of similar repeating subunits. Okay, so you see subunits are being repeated. For example, from carbohydrate down, that means carbohydrate, um, right, made up of monosaccharide. Monosaccharides are similar and they're repeating, right? Amino acids, right? Proteins are made up of amino acids which are similar and repeating. Nucleic acids are made up of nucleotides which are similar and repeating. So these ones are made up of similar repeating subunits are called polymers. This is very important. They are called polymers, right? And that means polymers include carbs, protein, okay, and of course, um, nucleic acids, DNA and RNA, right? So all of that, all right? So this is very important. So another important fact about biological molecules is that all biological molecules, of course, their subunits undergo what we call condensation reaction to form the bigger molecules, right? For example, if you have subunits always, right? The subunits undergo condensation. Now, this is usually reversible. When they undergo this first reaction called condensation, they form the bigger molecule, right? Or macromolecule. Do you get? So and of course, water is always produced during condensation reaction. So the bigger molecule can also undergo, all right, backward reaction, which is called hydrolysis, right, to give us smaller subunit, hydrolysis. Okay, all of them. Okay, very important. Okay, I'll continue the fact as we move to the next slide. All right, let's go to the next slide. Now, before I get into this, I want to talk about other important facts before I get into this glucose thing. All right, other important facts, for example, all biological molecules, right, their subunits are linked by covalent bond. Right? Yeah. So you see, amino acids in protein, amino acids are linked by a covalent bond called peptide bond. Peptide bond, right? So if you check in carbohydrates, the monosaccharides are linked by a covalent bond called glycosidic bond. Okay? Glycosidic bond. This is very, very important. Glycosidic bond. Okay? So glycosidic bond has two types. Most times can have more than one, but generally we have two. 1,4 and 1,6. Okay? So 1,4 is straight chain. So this is straight chain, and then this is branch chain. 1,6 is branch chain. Don't forget that. Okay? So let's go on. So we are starting with carbs. According to this we're starting with carbohydrates. Okay? So carbs, if you look at carbohydrates, they're usually made up of compounds that have carbon, hydrogen, oxygen, right? This carbon, hydrogen, oxygen, these compounds, they consist of two major functional groups, which can be, alkanal. So alkanal sugars are called, okay, aldose sugars. Aldose, all right? Some of them are made up of, called aldoses. Ketone functional group, they're called ketosis. Is that okay? All right. But basically, carbohydrates are divided into monosaccharides. Okay, you're going to find that monosaccharides, right? Monosaccharides, they are very, very important. They have one sugar unit. Mono means one, saccharide means one. Okay, so we have disaccharide, right? Disaccharide is made up of two sugar units in one molecule of the compound. We have polysaccharide. You can see polysaccharide consists of many sugar units. The word poly means many. Okay, the word poly means many. You can see that. Okay, the word poly means many. Okay, this word poly here means many. Okay, so if you come to monosaccharide, guys, I'm going to go back to this. You see, if you have monosaccharides here, monosaccharide. So monosaccharides um, have general molecular formula, GMF. Don't forget that, which is usually we call it, you know, put a bracket here, bracket C, right? H2O N. So where N is number of carbon atoms. Now this is very important. Carbon atoms. Okay. So of course, by the time you say if N is equals to 3, so just put 3 here. And if you put 3 out here, that means you're going to have multiply out, you're going to have C3, okay, H6 O3. That's a triose sugar, triose. Okay. Now if N is 4, it's going to be C4H um okay, let's deal with, okay, N equals to 5, yeah. It's going to be C5, yeah. Let's start with C5 H10 O 5. Okay. This this C5 H10, okay, O5 is a, a pentose. This is for pentose sugar, 5 carbon, see, right? So there are two of them. We have either ribose, okay, or deoxyribose. Deoxyribose, okay, deoxyribose, okay. So ribose is found in RNA and ATP. That's the pentose sugar in RNA, okay, and ATP. ATP. While deoxyribose is a sugar in only DNA. Okay. All right, that's it. Now they are called, so 5 carbon sugars are called pentoses. 3 carbon sugars are called trioses. What about 6 carbon sugars? Okay, 6 carbon sugars, what do you call them? All right, what do you call 6 carbon sugars? So just put 3 there. If you put 6 here, sorry, you multiply out. So that's going to give you C6H12O6. So these are for hexoses. Okay, 6 carbon sugars. Hexoses. Hexoses, guys. So example includes glucose, right? And not just glucose, we have fructose, okay, and then we have galactose. They are all hexoses, right? Why glucose and galactose? While glucose and galactose are aldoses, that means they have alkaline functional group. Fructose is a ketose, okay, right? So do you have any question, guys? Um, so that's it. Let's move on. Let's move on. So if you check what we have on the board here, we're dealing with glucose. If you check and see what it says, glucose has the molecular formula of this is a healthy sugar. Now is an energy source which is broken down calcium underlining during respiration. You can see it is also the monomer from which starch, so it starch cellulose are made. All right, there are two different kinds of glucose. Yeah, that's what we're going to look at now, right? There are two. Okay, we have alpha glucose and beta glucose, guys. And if you check, this is glucose, right? If you check, that's the what we call the structural formula of glucose. Okay, so this is the first carbon atom, the second, third, fourth, fifth, sixth. So this is the straight chain. So this particular chain here is a straight chain. Straight chain. So when straight chain, okay, undergoes some changes, right? The first carbon atom combines with the fifth carbon atom. that gives us a ring structure. Okay, so that's a ring structure. So if you check, this is a carbon atom 1 here, carbon atom 2 is here, 3 is here, okay, 4 is here, 5 is here, is the sixth one. Okay, sorry, carbon atom, yes, this is the sixth one. Yeah. So if you check the first carbon atom, the OH attached to that first carbon atom is below. If I draw the plane structure of this, the OH is below the plane structure. Okay, it's below the plane structure. All right, so we call it alpha glucose. OH attached to the first carbon atom is below the plane structure. Okay, is below the plane structure. Do you get? Is below the plane structure. All right, so yeah. So, but if you check this, this is still 1, 2, 3, 4, 5, 6. The OH attached to the first carbon atom here is above the plane structure. We call it beta glucose, all right? Beta glucose. So this is alpha glucose, and that's beta glucose. Okay? Very, very important, guys. Very important. All right, let's go to the next slide. So you see glucose structures, right? So you see glucose gives us this or that. So you see, this is the first carbon atom. The OH is below the plane of if you check this, follow this. This is the plane of the structure. This OH is below, right? Why this OH is above, right? Above what? The plane of the ring structure. So because it is below, it is above, we call it beta glucose. Because that one is no, because this is above, the one that's above is called beta, the one that's below is called alpha. Okay, above is called beta glucose. Do you get? So this is another way of drawing the structure of beta glucose. Come to the first carbon atom. Okay, that's the first, second, third, fourth, fifth, sixth. So the one attached to the first carbon atom is below. So this is below. So it also call it alpha glucose. The same thing here. 1, 2, 3, 4, 5, 6. So the first one, the OH attached is above. That's above the plane. It's above both of them. Okay, the other one is below the plane. That's what we call plane of ring structure. Plane of ring structure. Okay. So the one that is below the plane of ring structure is called alpha glucose. Alpha. So okay, that's the alpha glucose. That's the symbol. Why the one that's above the plane of the ring structure, the OH attached to the first carbon atom above the plane of the ring structure, that's above. We call it beta. Beta. Okay, glucose. So there are two isomers of glucose, either alpha or beta. Now I want to say this at this point, okay, that if we are dealing with galactose, remember that we have three major hexoses. Okay, so looking at their structure, we have talked about glucose, right? The structure of glucose. All right. Now, if we are talking about galactose has similar structure, similar ring structure as glucose. Both of them, they form what's called hexagonal ring structure. Ring structure. Okay. Yeah. So the only difference is that um, galactose is like alpha glucose, right? But the carbon 4, the OH attached to the carbon 4 is above. So galactose is just like this. If I should take it this way, look at it. Okay. Let me try to form galactose. All right. So this is CH2OH. All right. This is O. That's first carbon atom here. That's second. Okay. Sorry for it. Okay. So this is carbon 1. OH. OH. Yeah, H is here. This is OH down here. And carbon 2. We check carbon 2. OH is down. H is there. Carbon 3. OH is up. There. H is down. Carbon 4. 4. glucose in carbon 4, the OH is down. But for galactose, OH is up. Is that okay? Mhm. So that's the only difference between two of them. Okay? And well, this is H. So the only difference is this carbon 4. For galactose, the OH attached to carbon 4 is above like the plane or the ring structure. Right? But for alpha glucose, the OH attached to the carbon 4 is below. Can you see below? Is below here. This is above. Okay. All right. So these are glucose structure. Let's go to the next. So now this is knowledge check. Let's check what you got. Right. So structure shows which of them shows alpha glucose. Which of them shows alpha glucose? Okay. So let's try. So remember this is carbon one, two. Pause the video and try it. Three, four, five, six. So in alpha glucose, right, the OH group, OH attached, okay, to carbon one is below, remember, the plane, all right, of the ring structure. Don't forget that. They play on the ring structure. So now, of course, if you trace this now, the OH attached to this particular one, this is above, so this is not the answer. It's wrong. But if you check this is one, okay, trace it, is below, yeah, so this is the answer, right? So it's above here, all right? This should be OH, this should be there, right? So the answer here is the one that is below, which is option B. Okay, option B is the answer, guys. So the answer is B. All right. So you see what I said. See monomer. We're defining monomer. So what are monomers, guys? Read this with me. It's a monomer, simple molecules which is used as a basic building block. So all monomers are building blocks. Like monosaccharides are monomers. They are used to build disaccharide and polysaccharide. Okay. All right. So building blocks for the synthesis of a polymer. See, many monomers are joined together. How? How they join together? They're joined to make polymer, usually by condensation reaction. I was telling you about condensation reaction. And I told you there are bonds that are formed. You know, I mentioned protein, okay? That protein, the bond is peptide bond, but I never mentioned, right? I mentioned carbohydrates, right? When they undergo condensation reaction, you get, we have glycosidic bond joining the monomers, right? The, that form the polysaccharide. But I never mentioned nucleic acid. You didn't tell me nucleic 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 acid. Okay, the bonds that join them there is called phosphodiester bond. Phospho, yeah, phosphodiester bond. Now, if we're dealing with lipid, the bond that joins glycerol and fatty acid, the bond is called ester bond. Don't forget, right? So let's go on. Okay. So you see reactions example monosaccharides undergo conditional reaction to form polysaccharides. Amino acid undergoes, these guys form polysaccharides, right? Polysaccharides. These form proteins, right? Proteins. And these guys form nucleic acids. So, okay. But you know the bonds that join them together, covalent bond, right? So you see a polymer, let's go to polymer now, is a giant molecule made from monomers. Example, see, polysaccharide is a polymer, protein, nucleic acid. So macromolecules, they are just large and complex. Okay, they are formed due to polymerization of small molecules. Polysaccharides, nucleic acid. So any giant molecule is called a macromolecule, guys. Hmm? All right, let's go to the next. Okay, see monosaccharide, like I mentioned, monosaccharide, all right, is a molecule consisting of a single unit, single or one sugar unit. Hmm? So they form the simplex. You see that thing I did for you that time. This is the general molecular formula, GMF. Okay, that where we say that N is equals to number, right, of carbon atom. Remember? Yeah. Okay. So if N is 3, we call them trioses. So just put N here, 3 they are multiplied. If N was 5, if N was 5, Okay, we call them pentoses. All right, pentoses. Pentoses. If you put 5 there and multiply out, you're gonna have 5 times. You're gonna have C5H10 like we did. O5. Now this is ribose. Is a pentose. If you want to form deoxyribose from that, remove one oxygen. So you're going to be C5H10O4. So this is deoxy. Deoxyribose lacks one oxygen. Is that okay? Hmm? Lacks one oxygen. Yeah, it's found in DNA, right? So now, of course, is a disaccharide. That's disaccharide made up of two. It's a sugar which consists of two monosaccharides joined together by a glycoside. You see that glycoside bond? I told you that covalent bond. Okay. So if I have, for example, like disaccharide, they have glucose, okay, plus glucose. If both of them are alpha glucose and they undergo condensation reaction, we're going to have maltose, okay, and this and water, right? Maltose and water. So this reaction is condensation reaction. The backward is called hydrolysis. You know that, okay? So if, for example, you have glucose reactor, now this is alpha glucose, right? Alpha glucose. Now, if you have alpha glucose, okay, and galactose, right, undergo condensation reaction, they're going to give you lactose. Lactose is disaccharide. So disaccharide will have maltose. So maltose is a disaccharide, okay? Lactose, which is found in milk, is also a disaccharide made up of alpha glucose and galactose, right? Yeah, and if you have alpha glucose, see, alpha glucose plus beta glucose, right? Not beta glucose plus fructose. What am I saying? Plus fructose. Because plus fructose will give us sucrose. Yeah. So sucrose is also a disaccharide. There are two disaccharides. Okay. We have sucrose. So three: sucrose, lactose, and maltose. Okay. Sucrose is the form of carbohydrate transported in plants. Lactose is the sugar found in milk. That's the respiratory substrate for young mammals. Maltose is all right. Found is the respiratory substrate for germinating seed. You get? So if you want to find now, bring this down. If you want to find their general molecular formula, okay. So how do we find that? Okay. Remember that if I join two hexosugars, for example, C6H12O6 plus another C6H12O6, and then undergo that condensation reaction, they're going to give me, okay, if you write carbon, now sum of this carbon and that carbon, that's 12, C12. Sum of this hydrogen and that hydrogen, that's 24, right? C24, but remember that, no, that's hydrogen, sorry, it's hydrogen. Guys, remember that water is always formed, all right? So there's two hydrogen atoms in um, water. So minus, because if you say this plus this, right, that gives us 24 minus this 2, that gives us 22. So oxygen, 6 plus 6 is 12. 12 minus 1 oxygen water, that's 11. So for all disaccharide, this is general molecular formula for all disaccharide. So all disaccharide, they have 12 carbon atoms, 22 hydrogen atoms, 11 oxygen atoms. So whether it is this, okay, that's maltose or lactose or sucrose, okay? Now, but if we're dealing with polysaccharide, see, the word poly means many or several, many sugar units. So it's a polysaccharide whose subunits are monosaccharides joined together by glycosidic bonds. So there are many sugar units in polysaccharides, right? So polysaccharides, for example, we have starch is a polysaccharide. We have what else? Glycogen is a polysaccharide. We have cellulose. These are all polysaccharides. Okay, so polysaccharides are made up of many sugar units, many hexoses, right? So if you want to determine the general molecular formula of polysaccharide. Just take 1-hexo-C6H12O6. Put a number. This number means many of them, thousands of them, undergoes condensation reaction. Water will be formed. Remember, that's condensation. Now put, of course, bracket. We have 6 carbon H. Okay. Instead of 12H here, we have lost 2 to water. So it's going to be H10. Instead of oxygen should be 6, but we have lost 1 to water. It's 5. You put N here. So this is general molecular formula for all polysaccharides. Okay. C6 bracket N C6H10O5. Okay. So let's go on, guys. So another check. Is a trehalose. I want to pause the video and solve it. Is a sugar that gives negative result when tested with Benedict solution. So remember that any sugar that gives Benedict, okay, Benedict gives negative result to Benedict is usually a non-reducing sugar. If you watch the first video, okay, biological molecules factor, okay, non-reducing sugar, guys, right? So and then you know that is sucrose, okay. This is reducing. All monosaccharides are reducing. Fructose is reducing. But please don't forget that fructose is the only monosaccharide that form, right? It forms, what is it called? Pentagonal shape, right? Yeah. Others form, right? Hexagonal. Glucose and galactose form hexagonal, right? So only fructose that forms, right? So, but fructose is a monosaccharide, so it's also a reducing sugar. So we have non-reducing sugar, sucrose, sucrose. This is the only one that gives a negative result, right? And let's find out the molecular formula, right? What's the molecular formula for um sucrose? Sucrose is a disaccharide, right? So what's the molecular formula, guys? Hmm? I want to tell me the answer. Is it B or D? Okay, remember that it should be what? It should be B. Is that okay? How do we get that? Just combine two monosaccharide, two hexo sugar. Okay, C12H no C6H12 also like we did the other time, okay? Plus another C6H12O6. That will give us what? Okay, add up the carbon atoms. 6 plus 6 is 12. C12, right? Hydrogen. Remember that during condensation reaction, water is formed, okay? So let's add up hydrogen. 12 plus 12 is 24. H24, guys. No, but we're not going to write 24. We're going to minus 2, this 2. So it's going to be H22. Is that okay? Yeah, because 2 is already in water. So oxygen is 6 plus 6 minus this one. That's 6 plus 6 is 12 minus 1, 11. So you see that B is our answer. Okay. C12, H22, 11. Okay. So that is the general molecular formula for disaccharide. Okay. Sucrose, because it's a non-reducing sugar. All right. See, we mentioned these glycosidic bonds, covalent bonds that occur between, there's one between constituent monomer, and these monomers are monosaccharides, okay? And they are formed due to constitution reaction, which involves removal of water molecule in order to form polysaccharide, okay? And disaccharides, just sucrose. These constituent molecules can also be separated by hydrolysis. That's the backward reaction, okay? Which breaks the glycosidic bond between the monomer. For example, acid hydrolysis can be used to break them. Or okay, of non-reducing sugar sucrose, okay, bond, all right, in order to retrieve the constant monomer. Is that okay? So if I have, for example, sucrose consists of glucose, all right, alpha glucose plus fructose, two of them, right? All right. Of course, they combine together to give us sucrose. So if you break this bond, now break this bond, this is glycosidic bond, that's hydrolysis, okay? All right. But that's what joins them. So the reverse of condensation reaction is called hydrolysis. Okay, all right, it's called hydrolysis. It's called hydrolysis. Okay, it's called hydrolysis. So let's go to the next. Yeah, so you can see that, like I told you, that glycosidic bond has two types. We have 1,4. 1,4 is for straight chain. Straight chain, okay? So I want to show you how it is formed. Okay, how is it formed? So you see, so how is the 1,4 glycosidic bond formed? So you can see where it is formed. We're going to show you that. But just know that 1,4 is straight chain. And just know that 1,6 is for branch. This is 1,6 for branch chain. Okay, so you can see how 1,6 is formed. This one, you see the OH here on the first carbon atom here combines with, okay, hydrogen there combines with, all right, OH on the sixth carbon atom to form water. Okay, just like the way you normally do it. If you have this, I'm going to show you that this is that. Yeah. So if you want to form 1,4 glycosidic bond, this is OH down for alpha glucose. You have another alpha glucose here. Don't forget this is C here. Just sketching it. O here. This is another carbon, carbon 2, carbon 3, and they have OH here. So this H here combines with this OH here to form water. So this bond here is now, the bond is formed between this bond now is called glycosidic bond, but they call it 1,4 glycosidic bond. Why is it 1,4? Because this is first carbon atom, this is fourth carbon atom. Yeah. So, and because the alpha glucose OH is down, so call it alpha 1,4. Right? So guys, watch. You see that 1,4 is straight chain. Anyway, say 1,4 straight chain. Anyone want to form branch 1,6 branch chain? Okay. So 1,4 straight chain. You can say straight chain. All these are okay. Straight chain. Anyway, there's a branch. This is a 1,6 here. 1,6. That's branch 1,6 link. Okay. This is also a 1,6. Right. 1,6. That's branch. Now this is 1,4. Can you see 1,4 here? It's 1,4. Straight chain. Straight. Okay. All right. Now let's go to examples of disaccharide like what I've shown you. You can see what's going on here. This A. You can see that this OH here, this is alpha combines with this OH here. The hydrogen of here combines with hydroxide of this to give us water. This is alpha glucose. How do we know it's alpha glucose? Check the first carbon atom. Right, the first carbon atom here, OH is below the planar ring structure. If you're interested, okay, OH attached to the first one is below the planar ring structure. If you check the second molecule, OH attached to the first carbon atom, so this is alpha. So they are both alpha glucose molecules. So when they react, they undergo constant reaction. You can see the glycosidic bond here, right? So this forms water, and this is a disaccharide, and this is maltose. So maltose form between alpha glucose and alpha glucose. Okay. So if you check another one, okay, look at B, monosaccharide, monosaccharide, right? You see alpha glucose combines with beta fructose. Why is it fructose? Fructose because can you see is pentagonal shape? Okay, that's the only hexes that has pentagonal shape. But it has 6 carbon atoms. So that's 1, 2, 3, 4, 5, 6. Okay. So when they undergo constant reaction, they form glycosidic bond, and that's sucrose. So sucrose is made up of alpha glucose and beta fructose. Okay, so don't forget that, guys. Knowledge check. What cannot occur as a result of condensation reaction? What cannot occur? I want to pause the video and try it. It says breaking of glycosidic bond. So glycosidic bond is broken during hydrolysis. Okay, right? And hydrolysis cannot occur during condensation reaction. Formation of disaccharide is condensation. Okay, because they're joining two monosaccharides together. And what is from joining together two amino acids is condensation because amino acids are monomers. Okay, erosion of water molecule is also it occurs also in condensation. So the only one that um, cannot be condensation reaction is A. A must be hydrolysis. So the answer is A. So look at this. So which diagram? Now pause the video and try this. Try this out. Try it. So this is knowledge check. Try it out. So which diagrams show? See which diagrams show the release of water molecule during the formation of glycoside bond. So you can see number one. Let's check it out. You can see that this there's a reaction between He and OH of this to form water, and that's going to form glycosidic bond. It's going to be 1,4, right? So this is going to give us 1,4 length, 1,4 length. So if you check what is happening here, you see this is alpha glucose, what is rotated because oxygen is up here, oxygen is down here. There's a rotation, but guess what? Reaction is occurring also between H of this and OH of this to form water. So that's also glycosidic bond. Okay, it's still 1,4. Okay, because this is the first carbon atom here, and this is the fourth carbon atom here. So glycosidic bond is formed between them. Now, if you check number 3, you can see that the H here, okay, undergoes condensation reaction to OH here. And this is one carbon atom. This is the sixth carbon atom because this is the 1, 2, 3, 4, 5. So this is giving us 1,6 length. Okay. So which diagrams show the release of water molecule? All of them release water because it's okay. All of them. So A is the answer. Okay. So let's run. So see what I was trying to tell you. When two beta glucose molecules come together, especially in sucrose, there must be rotation of monosaccharide. Now this happens in sucrose. This happens in cellulose. Okay. Yeah, cellulose, right? So you see them rotating, but because if you look at it, okay, look at the beta glucose molecule. Let's see why this is very important. This is a beta glucose molecule, right? Yeah, how do we know? Just the first carbon atom here, which is always above. So if it's reacting with another beta glucose molecule, remember that this is carbon here. You can C here, OH, C, right? That's C here, C here, and of course OH is down here. So to move to bring this OH up here, right? This one will rotate, and that's what happens here. So it rotates by 180 degrees Celsius. So everything up here comes down, and that's what has happened here. This OH is up here, but it's down here. Okay, that's oxygen, right? And then of course this OH is up here, this is up here. So by the time it rotates, the OH comes up here. Is that okay? And then hydrogen on that combines OH of this to form water. Okay, this is called rotation. Beta glucose rotates 180. That happens. Okay, in cellulose, there's always a rotation 180, so that glycosidic bond can be formed, guys. All right, look at this. So the diagram shows part of the chemical structure of naturally occurring polysaccharides. So what type of glycosidic bonds are present? So I want to check out what type of glycosidic bond. Now, if you check, you see that the OH of this post video and try it, the OH was up. So this is out. This is beta. The OH attached to the this first carbon atom, second, third, fourth, fifth, and this is sixth. And if you check this one also, 1, 2, 3, right, 4, 5, 6. So the OH here, so this first one is beta glucose. So if you check, the OH here is also up, right? So this is also beta glucose, right? But you see, um, there's a combination between first carbon atom here. So these two will give us 1,4 lengths, right? This is number 1, this is the fourth. But if you come here, okay, if you check, this is number 1 here, it will be 1, right? No, this is 1, okay, this is 3, yes, because this one is between the third carbon atom here and the first carbon, so it's going to be 1, 3, right? 1, 3. So this will be 1,4 because this is first and the fourth. right? First and fourth. So that means it's going to give 1,4. So it's going to be beta glucose. So anything alpha cancel. Beta, yes, it's going to be beta 1, 1, 3, and 1, 4. This is 1, 3, 1, 4. So the answer is C. That's my answer. Okay? So that's I C. So it's a monosaccharide. So you see that I showed before glucose reacting with fructose. How do I know fructose? Fructose is a hexose, but it has pentagonal shape. It has, five-sided shape. Okay? But, glucose has hexagonal, right? So when they combine, this is, all right, condensation, backward is hydrolysis. So you see what is happening, water is being formed, and that's glycosyl bond, and guess this is 1, 2 glycosyl bond. Okay? So this is the first carbon atom here and the second carbon atom here. Okay? Yeah. So that's 1, 2 glycosyl. Okay, bond. Right. So you see, so that's all about that. Let's come to polysaccharides. So one of them is starch. So starch is a macromolecule that is found in plant cell. Okay. So one thing is very important. Now, when you are dealing with starch, starch is a storage molecule. Storage molecule is a polysaccharide. Okay. Starch is glucose store. Glucose store. Very important. Glucose store. But in plant cells. Plant cell. Okay. And remember, for all biological molecules, you must know that there's something called structure. All right. Structure determines the property, and the property determines the role or function. So the role of starch is storage. So this is the role. Starch stores glucose in plants. So the question is, um, let's look at the structure of starch. So don't forget that starch, the monomer is alpha glucose. So you look at the structure, right? Alpha glucose make up starch. And then starch is a polysaccharide of two other polysaccharides. So when you look at starch, starch made up of two other polysaccharides. One is called amylose. Amylose is a polysaccharide of alpha glucose. The other one is called amylopectin. Okay, so amylose is unbranched. So because the polysaccharide is unbranched, it's going to have only 1,4 links because it's not branched. Amylopectin is branched. It has few branches. So it's branched. It has few branches anyway. So it's going to have more 1,4, but also it also have um it also have 1,6, right? Because you have few branch. Okay, is that okay? So that's the structure of starch. Now, because of that, starch has some property. For example, starch is insoluble. So because starch is insoluble, there's no effect on the osmotic property, no effect on the osmotic property. Is that okay? Property of the cell. So it does not allow water flowing out and water flowing into the water, okay, into the cells, okay. So starch is also because it's a property. So starch is bulky or large, so it cannot leave the cell. Very important. Starch, um, this is key. Starch has compact structure. You know, compact structure means it can store more glucose units in one more glucose unit, right? In small space. That's compact structure. Okay. Yeah, very important. We're going to discuss many of that. So starch is highly compact. You can see that we mentioned it here, right? Stores energy. So you check this. So this is amylose. This is amylopectin. You see, this is structure. So amylose has only 1,4 because it's unbranched. Okay. So amylopectin has both 1,4 and 1,6 because it's branch. See, branch structure. So amylose is helical, right? It's a branch. Okay, it's helical. This is also amylose is spiral, and amylopectin is compact, right? So this is amylopectin. See, it has branches. Okay, that means it has 1,4 link and 1,6, but it has more of 1,4. So you see amylose. So amylose, right, is just helical. It's unbranched. It's unbranched. You can see that. You can see that it's unbranched. But you see, their similarity. The similarity is that both of them are made up of alpha glucose. Okay, alpha glucose monomer. All right, you get so and both of them have 1,4 links. Right, but if you check their differences, okay, this amylopectin is unbranched, right? Sorry, amylopectin is branched. What am I saying? It's branched. Therefore, it has 1,6. But this guy is not branched. Okay, it's unbranched. That's amylose. He has only 1,4. Okay, and while this is helical and spiral, this is also compact. Okay, guys, let's go to the next page. You see, um, this is glycogen. Glycogen is also a storage molecule. It's glucose store. So glucose store in animal cell. Okay, yeah. So you can see that a macromolecule that is used to storage an animal cell. You can see that goes to an animal cell, and it's also made up of alpha glucose molecule. Yeah. So you see, the monomer is alpha glucose. Okay, you get. So glycogen is very similar to that of amylopectin. However, it is more branched and therefore contains more 1,6. Okay, because anyway, you see branch, there will be 1,6. So glycogen has more 1,6, more 1,6. Okay, all right. Now, but you see, like I told you, there's a place for structure and then there's a place for property and there's a place for role. Anytime you are dealing with biological molecules. So the role of glycogen is that it is glucose store. Okay, yeah. Okay, let's link it to the structure. The structure is made up of alpha glucose. Okay, so it's made up of alpha glucose. It's highly branched, highly branched. Okay, highly branched. But if you check the property is that, of course, like starch, it is insoluble, so it does not affect the osmotic property of the cell. It is compact. It can store many glucose units in a small space, right? It has many free ends. So it has many free ends or terminals. Okay. So now because of that, glucose subunits can either be easily joined or removed. Glucose subunits. Okay. Don't forget that. That is also bulky and cannot leave the cell. Right. But let's look at another. This is another very important um polysaccharide called cellulose. But you see, it's also found in cell wall of plant cell and is made up of okay, beta glucose subunit that forms beta glucose. The 1,4 glucose bonds. Alternate beta glucose molecules are rotated by 180 degrees. Okay, in order to form these bonds. Very important. So you see, if you discuss now cellulose, so if you look at the role of cellulose, if I ask what's the role of cellulose, you can say that cellulose is simple, is uh, a structural polysaccharide. Structural polysaccharide. So polysaccharide. What does that mean? It it gives strength and support to cell wall. Strength and support. That's one of the rules. But also has a rule, and that rule is that it also allows transport of water by apoplast because it's polar. Okay, but you see, when you look at the structure of cellulose, which is what we started here, the cellulose, the monomer of cellulose is beta glucose. Don't forget that monomer is beta glucose. Okay, now it's unbranched, right? So beta glucose, and then the beta glucose molecules, like we said, they're always rotated, right? There's a rotation, like I showed you, rotated by 180 degrees Celsius. Okay, so don't forget that when we're dealing with cellulose, okay, cellulose all right, has what we call cellulose molecule. We're going to discuss that. It has cellulose fiber, okay, right, and then cellulose microfibril. So cellulose molecules, they are made up of beta glucose, okay, made up of beta glucose. Do you get? So in cellulose molecules, we're going to look at it. You see beta glucose, all right, are rotated by 180. So if you're asked, you see that we can say that cellulose molecule, all right, basically, guys, you see this cellulose molecule, all right, is on branch chain. So the first thing you say is on branch chain of beta glucose, in which adjacent beta glucose is rotated by 180, right? Okay. So let me show you that translate. So we're gonna see that. So if you have, I'll show you how it is rotated, but you know, is on branch chain, on branch chain. So there's a rotation here. Oxygen is here, oxygen is down here, oxygen is here. Okay, oxygen is there. There's okay rotation. So if this is and then very importantly, you have to know that that rotation helps to cause hydrogen ion, hydroxyl ion project, because they can ask you that project in different direction. Okay, different direction. Mhm. So, all right, and what is the role of this? The role of this is that it helps to form. The first role is that it helps to form hydrogen bond between cellulose molecules. So a cellulose molecule just consists of um, chain on branch chain, right? So now this is molecule one, this is molecule two. Because of rotation, hydrogen bond can be formed between them. Okay, hydrogen bond formed between parallel cellulose molecule. Okay, now parallel cellulose molecule. That hydrogen bond gives cellulose a property called high tensile strength. High tensile strength. Okay, high tensile strength is why glucose is resistant to stretching. Resistant to stretching, right? Or it can withstand large pulling forces. So it withstands large pulling forces. Okay? And that's why it's able to give the role of structure, like it gives support and strength to the cell wall. So if we're dealing with, let me tell you this. So we always have role, we always have property, okay, we always have structure. Okay. So they ask to connect role to structure. For cellulose, the role is that it has two roles. The first role is that it provides support and strength, all right, to cell wall. Mhm. And the second role is that it also helps for water transport through cell wall. So now, why does it offer strength and support? They come to property because it has high tensile strength. Okay? High tensile strength means that it is resistant to stretching and can withstand large pull forces. Now, why does it have high tensile strength? Because of hydrogen bond. Now, hydrogen bond between cellulose, parallel cellulose molecules. Okay, are you there? Yeah. So this hydrogen bond is formed because cellulose molecules consisting of um, adjacent, of course, on branch beta glucose. So this is beta glucose, but rotated by 180 degrees Celsius. Okay. So now, but if you follow now, that rotation causes OH to be projected in different direction, and then can form hydrogen bond. That also rotation causes cellulose molecules to become polar and hydrophilic, and that's why they can allow water transport. Okay. So look at this. Okay, that's glycogen I told you about. See, glycogen, it has many 1,6 link, 1,6, okay, it's highly branched. Okay, 1,6, 1,6, that's it. Right? You see that rotation I told you about? This has to rotate, okay, in cellulose molecule. So cellulose molecule, after rotation, this is cellulose molecule. Can you see? So cellulose molecule consists of and this is molecule one, molecule two, three, four. Can you see that? So these molecules are they're linked by beta 1,4. All right, beta 1, beta 1, 4 links, okay? They're unbranched, is that okay? And there's a rotation by 180. That rotation, like I told you, projects OH in different directions, okay? Okay, so you see, unbranched beta glucose molecules linked by beta 1,4 glycosidic bond is the cellulose molecule. Do you get? And then many molecules linked by hydrogen bonds will give us fibril, microfibril. Okay, so we're going to see that right now what it is. But before then, let's answer this question. Which role is correct for cellulose? So rotation of, pause the video and try it. Rotation of 180 monomer by 180. Yeah, that's correct. So A and D, they are suitable answers. Shaped of molecule. Is it branched? No, cellulose is unbranched. So that means D is leading now. Hydrogen bond between molecule, yes. So the answer is D. Okay. Now look at what I was trying to say, guys. Right? This entire thing here, you see this entire thing here. Okay, what do you call it? We'll call it cellulose fiber. So you see that molecule, cellulose molecule, right, makes up microfibril. Microfibril makes up fiber. So the largest is fiber. So this is fiber. See, cellulose fiber. Cellulose fiber made up of many. See, cellulose fiber. See what they tell them? Cellulose fiber made up of many microfibrils. So when you come to a microfibrils, see a microfibril is made up of 60 to 70 cellulose molecules. Can you see that? Now these 60 70 molecules, so this is cellulose, right? Fibril, microfibril, right? Right? Made up of cellulose. So each chain here, one chain here is called a cellulose molecule. Okay, so molecules running parallel to each other. They run parallel to each other. They are linked by this hydrogen bond, H bond. Okay, this bond here, hydrogen bond. Okay, that bond here, right? Hydrogen bonds. All right, join. That's how you can see that hydrogen bond. Join cellulose molecules, right? About 60 to 70 of them to form microfibril, right? And many microfibrils joined by the same hydrogen bonds form cellulose fiber. Okay. Now let's check. Which statement describes features of cellulose that adapt it for its function in plant cell? Yeah. So what feature adapt it? All right. Pause the video and try it. Number one um number one says three cellulose molecules coil around each other to form a triple. Do they form a triple helix? Let's check. Do they form cellulose molecules? Do they form triple helix? They don't form triple helix is something like this. I see. But that's not so with this. It's not so with this. Okay, so that means this is wrong. Number two says main hydrogen bonds form between adjacent cellulose molecule. Yeah. Go back there. You see, this is a molecule one. This is molecule two. Can hydrogen bonds form? See that this hydrogen bond? Okay. Yeah, that's correct. So this is correct. Number two, covalent bonds form between adjacent cellulose molecule. Now the only thing that is formed between them is only hydrogen bonds, right? So that's wrong. The answer is two only, which is D. All right. So you see, now let's check again. So the diagram shows three hexoses. The three hexoses. Okay. That means hexose means they're made up of six carbon atoms. One, two, three, four, 5, 6. Check it out. 1, 2, 3, 4, 5. This first carbon 6. Okay. Check this one. 1. This first carbon 2, 3, 4, 5, 6. They're all headless. Okay. 6 headless. So then answer which row correctly shows examples of carbohydrates in which these three hexoses sugars occur. Remember that sucrose, all right, sucrose is made up of fructose, all right, plus alpha glucose, all right. So sucrose must have fructose. So this is fructose. How do I know? This number 3 is fructose. How do I know? It has a pentagonal shape. Even though it's a hexo sugar, but it has pentagonal shape, 5-sided. So 3 must be there for fructose. Okay? It's not here. It's not here. Of course, yeah, if you okay, alpha glucose should be there, which is 1. Yes, but let's check. In cellulose, if you check, let's even check. This one is what? If you create a plane around the plane or the structure, you see this OH is below the plane. What is that? That is alpha glucose, the only alpha glucose. You take the plane around the plane of the structure. The OH attached to the first, this is first carbon atom, OH attached to this right? That is beta glucose. So cellulose movement to beta. So this goes for 2 is here 2. Okay. Of course, amylopectin, alpha glucose. So it's 1. 1 here, 1 here. Okay. So the answer is D. Because 3 is fructose. It must be in sucrose. Okay. Sucrose is made up of fructose and beta glucose. Cellulose is beta. So this 2 is cellulose. Okay. See that this is found in sucrose, right? And this one, so D is the answer. Right. So now we're doing triglycerides quickly. So wrap up. So triglycerides, tri. So triglycerides is a type of lipid. Remember, we're now doing lipid. So in AS lipid, we're gonna deal with triglyceride and phospholipid. Okay, phospholipid, phospholipid. You have to master 2 of them. So triglycerides consist of right three fatty acids, 3 fatty acids. Don't forget that. Okay, and 1 glycerol. Do you get? But in phospholipid, we have 2 fatty acids. So one of the fatty acids, is displaced by a phosphate. 2 fatty acids, we have glycerol, 1 glycerol, and 1 phosphate group. So if you are asked for the similarities between them, on triglyceride have between triglyceride and phospholipid, both of them have fatty acids, okay, and both of them have glycerol, right? Now remember that I told the lipids consist of fatty acid and glycerol, especially of course triglycerol, right? But they are linked by ester bond, okay? So glycerol is linked by ester bond to each um, fatty acid. Okay. So because if you look at these guys, now I'm going to show you that later. But this is glycerol. Okay. This is fatty acid. Then the fatty acid there, fatty acid there. So each one, this is where we have ester bond. So it has 3 ester bond for triglyceride. But if you look at this guy, phospholipid, he has only 2 ester bond. Okay. Glyceride has 3 fatty acid. 1, 2, 3. That means 3 ester bond. This has 2 fatty acid. 1, 2. Let's go back. Okay. 1, 2. And therefore 2 ester bond. Now that means that when this is formed, three water molecules are formed. These 2 water molecules are formed. Okay. We're going to get into that. But just as a word of introduction, just know that, all right, triglycerides forms by constitution of 3 fatty acid chain and glycerol molecule. 3, right? Fatty acid 1, 2, 3, forming an ester bond. Fatty acid chains along hydrocarbon chains with carbonyl head. Glycerol is an alcohol contain 3 OH group. Don't forget that. I'll show you that. Now, unsaturated fatty acid, when it's unsaturated, that means they have less hydrogen atoms and usually carbon bond or triple bond. Okay, the more of this double bond and the more unsaturated. We call it polyunsaturated. More than one is saturated to polyunsaturated. So saturated contains only single bond, right? That are solid at room temperature. Right? Look at this. Let's see what I want to show you. Now, during the formation of this is called condensation reaction, right? For triglyceride. So this is glycerol. You can see it has 3 OH, 1, 2, 3. And then it's combining with 3 fatty acids, 1, 2, 3. For each one, okay, this hydrogen here forms water with OH here. See, so they're gonna form 3 water molecules. Okay, so this is water, that is water, that is water. Okay, and then we have triglyceride. So this is an ester bond. This is ester bond. Ester. That's ester bond. So yeah, three ester bonds. That's ester. Okay. So, but if it is phospholipid, you have 2 ester bonds. Okay. All right. So 3 water form. So now that's good. If you go backward, that's hydrolysis. You have 3 fatty acid. Okay. 3 fatty acid and the last. Okay. So another check. What is the maximum number of water molecules produced when a triglyceride is inside? What's the answer? Because try to read the answer. That's a 3. You have 1, 2, 3. Okay. So the answer is 3. But if they change it and they put phospholipid here, the answer will become 2. Phospholipid 2. Okay, look at this question. The table shows the number of carbon atoms and the, now I want you to pause and just try to solve this question. Okay? The table shows the number of carbon atoms and the number of, yeah, you see carbon atom first and then carbon to carbon double bond molecules of 3 triglyceride and fatty acids. Okay, so we're dealing with 3 triglyceride and fatty acids. The formation is shown in the former number of carbon atoms. So number of carbon atoms is to number of carbon to carbon double bonds. So this triglyceride, right? Okay, see that's triglyceride, fatty acid one, fatty acid. So in every triglyceride, we have fatty acid one, just like we have like this. Okay, this is fatty acid one, this is fatty acid two, this is fatty acid three. Yeah. So now this triglyceride, fatty acid one, two, three. So guys, let's go. So if you check now, triglyceride, remember it is number of carbon atom, like they said, that's the ratio, and number of carbon double bond. So the question says, which description matches fatty acid 2 or the 556 is to 4 triglyceride and 5 in table as X, please. Okay. So remember in triglyceride here, okay, if you check the total number of carbon atom is presented because triglyceride, watch this guys, total number of carbon atom will count all the carbon atoms here, all the carbon atoms here, all the carbon atoms here, including that, and then count these 3, 1, 2, 3 of glycerol to get it. You get? So if you check this, to get 56 will be 17 plus 18, right? Okay, of course, the remember 17 plus 18 is what? Let's tell me that's what. 7 plus 8 is 15. That's 15, right? Right, 5. Put 1 here, that's 35, right? Okay, so to find the other carbon atom, right? Remember that we already have three for glycerol. Glycerol is three, right? So we have this for first fatty acid, fatty acid, fatty acid, one, fatty acid, two. Okay, let me put three for glycerol. So if you put three there, that's going to be what's 38, right? So if you want to know the one for fatty acid, three. So that means the total, which is 56, is 56, right? Minus 38. Okay, it's going to be 8. 5 minus 5 minus, that's going to be 18. Okay, so that means that for X, the carbon atom is going to be 18 is to how many bonds does it have? Remember, we have 4 double bonds here. Out of these 4, fatty acid 1 doesn't have any. So fatty acid 3 has 1. So fatty acid 2 should have 2 to balance it 4. That means 18 is 2, right? 2, right? And 2 means that it is unsaturated. It has double, okay? So it has 18 um, carbons for that fatty acid here. It has 18, right? And then 2. Right? So 2 plus 2 gives us 4. 17 plus 18 plus 18 plus 3 times 1. I think is 3 carbon plus 4. That gives 6. Then if it's unsaturated, it cannot be saturated because then it has double one. It is not 17. It is 18 unsaturated. That's the answer. So see, rule of triglyceride, you can see they're long-term energy stores, right? Why? Because they have more carbon hydrogen bonds or more hydrogen atoms. Okay. Now, triglycerides release more than twice amount of energy compared to equivalent mass of carbohydrates. Okay, because it has more hydrogen atoms or more carbon. So it acts as insulator, provides buoyancy, acts as insulator to prevent heat loss because the poor conductor of heat. In aquatic animals, it provides buoyancy so that aquatic animals can swim. The blue whale has fat deposit called blubber. Blubber. Blubber is a fat deposit that allows it to swim. Now, metabolic source of water, right? So phospholipid molecules, hydrophilic head, is the difference between it and triglyceride. It has a hydrophilic head. It has hydrophilic head and hydrophilic tail, right? It has a phosphate group. It has two fatty acids. You can see that why hydrophilic tail contains two fatty acids. This is due to partial negative charge on the phosphate group that gets attracted to partial positive charge in water. So you can see that phospholipid has a hydrophilic head. Let's see. You can see that. So that's hydrophilic. So if you compare it with triglyceride acid, so triglyceride has three fatty acids, right? This has two. That means three ester bond. There's two ester bond. Okay. Three water form, two water form. Okay. All right. So if you check again, this has phosphate, no phosphate. Okay. All right. So you can see the role of phospholipid. Cell membrane usually. Yeah. So this is where we'll stop. But let's check this. Look at this knowledge check. What causes the phosphate heads of phospholipid to become polar? The phosphate head positively and tried saturation. The phosphate heads are joined to water molecules by hydrogen bonds. What makes it polar? Is it hydrogen water? No, it is because it is polar. That's why it can join with hydrogen bond. Okay, so it says the phosphate heads are insoluble in water. No, they are soluble. Okay, see, it says the phosphate head become ionized in water. Yes, that's why they are polar. And these phosphate heads are joined to water molecule by covalent bond by hydrogen bond because ionized in water. That's why they're polar. Look at this. Which statement about triglyceride is correct? I want to pause it and look at it. In any triglyceride, all the fatty acids are saturated, or all the fatty acids, no, it can be a mixture of them. All right. two triglycerides are joined together by an ester bond. In triglycerides is three fatty acids, right? Joined to one glycerol. So that is wrong. So it says triglycerides are polar, but they're not. They're non-polar, right? It's phospholipid that has a partial polar part. Glycerol is joined to each fatty acid by covalent bond. Yeah, that this covalent bond is an ester bond. Ester bond. Okay. So D is the answer. Right. Thank you for sticking to us. Kindly watch out for the next part and leave your questions. Of course, the next part is biological molecules part 2, part 3. We're just done. Please like, subscribe, and turn on the notification button on that investor. All right. See you guys. Bye. Okay. So remember this issue.